This video is about the specific immune response, part of the section on cell recognition and the immune system. You've already looked at the non-specific immune response. You've seen that it is antigen independent. It doesn't matter what sort of antigen it is, the response is exactly the same. There is also an immediate maximal response. As soon as a pathogen is detected, inflammation, localized temperature increase or fever, and edema or swelling will occur. It isn't antigen specific, so there is no directed response to a particular type of pathogen, and the exposure to that pathogen results in no immunological memory from just the non-specific response. However, the specific immune response is antigen dependent. You may see different amounts of the antibody-mediated versus cell-mediated response depending on which sort of pathogen it is. There is also a lag phase between the initial exposure and maximal response, and we'll see that when we look at how immunity is acquired through suffering an illness and through vaccination. The response is also antigen specific, so it will target each newly encountered pathogen the same way on first inspection. The exposure also results in an immunological memory. We will retain our memory cells within our immune glands, and they will be released very rapidly to bring about a more immediate response on a second exposure. There are two types of white blood cells, specifically lymphocytes, involved in the specific immune response. The first are the B cells. You'll note the cute little B at the bottom of the page. This is to help you remember them. The B cells interact directly with the antigens, so they have receptors on the cell surface membrane that are able to directly bind in a complementary fashion to the antigens in the blood. And they produce and secrete antibodies. So the activated B cells differentiate into plasma cells with large numbers of ribosomes and rough endoplasmic reticulum. This is because the antibodies being produced are protein-based. So these plasma cells just keep on churning out antibodies after antibodies. Now, some of the activated B cells will remain in our circulation as B memory cells. Then there are the T lymphocytes, and again you will be delighted to note the cup of tea at the bottom of the page to help you remember. The T lymphocytes can only detect the antigens when they are presented in these cell surface proteins called MHC proteins. That stands for major histocompatibility complex proteins, but that's not going to be something you'll be expected to recall on the exam. Now, T lymphocytes differentiate before they mature, whereas the B cells that we saw differentiated after they were already mature and in contact with the pathogen. So T lymphocytes are divided into T helper cells, T killer or cytotoxic T cells, T memory cells and T regulator cells. Let's look at what each of these does. So in B cell activation, let's look at the summary. There is a humoral or antibody-mediated response. This comes from the old idea that there were 4 humours in the body and the blood was one of them. So it was believed that this immune response went through the humours. Thank goodness for 21st century science. So the B cells differentiate into the plasma cells, antibodies are secreted. That's why it's sometimes better just to refer to it as the antibody-mediated response. And then we have T cell activation. So that is the cell-mediated response. The T cells become cytotoxic, so they are toxic to cells, and they will directly attack the invading antigens. These MHC proteins are the major histocompatibility complex, as I said earlier. So any infected cell or a phagocyte, so that's going to be a macrophage or a neutrophil, will present any foreign antigens on these MHC proteins, and then we refer to it as an antigen presenting cell, or an APC. So let's look in more depth at the role of each of the B cells. So plasma cells will circulate in the blood once they've been activated, and they are just going to manufacture and release antibodies. So there's going to be a massive amount of protein synthesis going on, lots of mitochondria expected, lots of vesicles, Golgi apparatus, and the endoplasmic reticulum that we referred to earlier. The B memory cells, on the other hand, will remain in the body for several years, and they will just act as an immunological sort of memory. So they will retain the precise codes for those specific antibodies and be able to very rapidly manufacture them. Here are the T cells that we are going to look at in the immune system. So the T helper cells release molecules called cytokines. Now these will activate or move cells, and they stimulate the B cells to develop, and they also will stimulate phagocytosis to occur. Then we've got the T killer cells, or cytotoxic T cells, which will attack and kill any host body cells that display the foreign antigen. So a lot of what the immune response does here is attack our own infected cells. I put these down at the bottom in italics because you do not need to know these ones for the exam. There are also T memory cells that will help with the provision of long-term immunity. So they will remember the precise killer cells needed. There are also T regulator cells which can shut down the immune response after the pathogen has been removed. There's no point in carrying on fighting a pathogen that isn't there anymore. So this helps the body to retain essential resources. So let's have a look at my wondrous animations for the antibody-mediated or humoral immunity. So first off, we've got an antigen-presenting cell. That's the bright yellow cell. And that will bind with the T helper cell, which is shown here in pale blue. And that activates the T helper cell and triggers the division of the T helper cell. So we'll get some memory cells, and most of them will be other T helper cells. So this stage is known as T cell activation. Next, we see that a B cell can also become an antigen presenting cell, and the activated T helper cells bind to the antigen presenting cell and produce cytokines. These ones are called interleukins. That just means between the white blood cells. The B cell then divides to produce B memory and B effector cells. So this is called clonal selection. You don't need particularly to worry about the B effector cells, but they are the ones that differentiate into plasma cells. So in that respect, they are the intermediate, but you're not going to be expected to recall them. These plasma cells will then be secreting antibodies specific to the original antigens. And the plasma cells are identical to the B effector cells, and so this is referred to as clonal expansion because they're clones. Let's look at the cell-mediated response. The T killer cells, the cytotoxic T cells, bind to an antigen-presenting cell. And there are interleukins from those T helper cells that were produced in the previous slide, which stimulate that cytotoxic T killer cell to divide. So we have clones of the T killer cells, and then some of those differentiate into T killer memory cells. The T killer cell binds to the infected cell and lyses it using a molecule called perforin. So here's a scanning electron micrograph of a white blood cell, or lots of white blood cells rather, crowding around a cancerous cell. The T killer cells that you can see bind to the MHC on that cell surface. Interleukin-1 from an antigen-presenting cell will stimulate the division of these T killer cells, and they release perforins, which make pores appear in any of the infected cell surface membranes. So that means hydrogen peroxide and nitric oxide enter the cells. Neither of those are particularly good for the old cell, and they swell up and burst, leaving behind respiratory substrates and so on that could be used and recycled in the body. So that concludes this short video on the specific immune response. You can go back and review these slides and pause it at any time if you want to make additional notes, but you will be expected to be able to discuss these stages in the immune response in your next lesson.